Hello everyone, my name is Sharon. Today I'm going to talk about effects of salinity changes on castor, Antarctic, phytoplankton, physiology, and assemblage composition. And as first, I will tell you what's special about Antarctic seawater. Look at this global temperature data. We can see that the global average temperature rises very fast in the recent years. And that means our Earth becomes warmer. And that's a serious problem we have to face. However, we all know that Antarctica has the coldest climate on the Earth. So we want to know how global warming affects Antarctica. Because of global warming, main air temperature have increased significantly over the past 50 years. And these long-term consequences on sea ice and seashell dynamics, as well as glacial melting, and cause the freshwater input into the Antarctic seawater. So salinity of Antarctic seawater must be lower than normal. And in this study, Antarctic seawater is regarded as low salinity treatment, we call LST, and compared with normal salinity treatment, we call NST. And the author collected the Antarctic seawater at about 5 meters deep in the polar core, and the yellow stuff shows the polar core. And what's wrong with phytoplankton in this low salinity treatment? And look at this picture. And when the cell changes the environment from normal salinity to low salinity, and the water will influx into the cell, then the cell will full of water gradually. So phytoplankton suffer from hyperosmotic threat that result in increased oxidative stress. Also effects on a variety of metabolic pathways. And what is oxidative stress? It will occur when reacting oxygen species formation exceeds antioxidant defense capability or disrupt redox signaling, and has been linked to many cellular toxic processes, including damage to protein, enzyme inactivation, and DNA breakage. And what is reacting oxygen species? We call ROS. And they are formed as a byproduct of normal metabolism of oxygen and have important roles in cell signaling and homeostasis, and such as superoxide anion, peroxide, and hydrogen peroxide. And our objective was to experimentally characterize the response of the natural phytoplankton assemblage from the castor Antarctic environment that is increasingly subject to changes in salinity. And to reach this goal, we studied phytoplankton assemblage biomass, composition, growth, and physiology. And physiology includes oxidative stress, photosynthesis, and respiration. And how did we get the data? First, we measured chlorophyll A concentration to obtain the assemblage biomass. And second, to know the composition, we did cell count by microscope. Third, the author set an equation to calculate the growth rate. And this is the equation. The most important is physiology. The author focused on oxidative stress, such as cellular oxidation and lipid peroxidation. And lipid peroxidation means free radicals steal electrons from the lipid in the cell membranes, and it may cause cell damage. And in the case of cellular oxidation, we measure dichlorofluorescent diacetate, DCFHDA oxidation rate. Because when DCFHDA enters the cell, it will be hydrolyzed to become DCF, and will be rapidly oxidized to become DCF, and which is a highly fluorescent compound, so that we can trace the oxidation rate easily. And the other case is lipid peroxidation. We measured 2-thiobarbitric acid reactive substance, TBARS concentration, which were used as an indicator of RS-induced lipid peroxidation. Then we also concern lipid cellular antioxidant, alpha-tocopherol and beta-carotene. We call them alpha T and beta C, and so that we know the defense capability. Next is photosynthesis and respiration. We measure dissolved oxygen concentration in the seawater. Then see what we attain. And in these two pictures, the x-axis are exposure days, and the y-axis in picture A is chlorophyll A concentration. In picture B is abundance. And we can see the trend of chlorophyll A concentration is similar with abundance. And that is the more chlorophyll A, the more phytoplankton assemblage. And we also can see the readings of NST show higher than LST. And under the microscope, we discovered that diatom were the most abundant organism. And in normal salinity treatment, the abundance of large century diatom continue increase during the whole experiment. However, in low salinity treatment, we observe large century diatom species during the first 2 days, but change to small pennant diatom species from day 4 to the 8th. And this is the large century diatom, and this is the small pennant diatom. And in this picture, the x-axis is exposure days, and the y-axis is growth rate. And we can focus on day 4. After day 4, the growth rate increased in LST but decreased in NST. So that means osmotic thread inhibit phytoplankton growth rate and change their composition. And come to oxidation. And in these two pictures, the x-axis are exposure days, and the y-axis in picture A is DCF-HDA oxidation rate. In picture B is TBARS concentration. And we can see the readings of LST show higher than NST. And then we can know phytoplankton suffer from more oxidative stress in low salinity treatment. In these two pictures, the x-axis are exposure days, and the y-axis in picture A is alpha T concentration. In picture B is beta C concentration. And in yellow box shows the structure of alpha T, and in the green box shows the structure of beta C. And we also can see the readings of LST show higher than NST. And that means protective role of alpha T and beta C are strongly against hyperosmotic induced damage. And last is photosynthesis and respiration. And in picture A, the x-axis is exposure days, and the y-axis is oxygen production. And in picture B, the x-axis is exposure days, and the y-axis is oxygen consumption. And there have a significant difference during the first 4 days. And that can prove changes in salinity can strongly influence Antarctic phytoplankton, photosynthesis, and respiration. And come to conclusion. And combine all the effects. Response to hypotonic stress of different severity may link to species-specific and exist the possible of species-specific mechanism of osmotic acclimation. And species-specific means this species can do something but another species can't. And acclimation means An organism adapts its natural climate environment after being some changes. Thank you.